Hey group, and welcome to Medical Microbiology 1. We're going to talk about a lot of great things this semester, and let's go ahead and start with Unit 1, which is safety in the microbiology laboratory. There are many roles that the microbiologist plays when they are working in a microbiology laboratory. One such role is called the diagnostic role, where we identify organisms and provide susceptibility testing. That's probably one of the most important things that we do in a microbiology lab. We not only tell the physician the name of the organism that we've isolated, but we provide information on what antibiotics they can use to treat that. We identify infectious organisms and determine which antibiotics can be used to treat those organisms or infections. Another hat that the microbiologist wears is the epidemiological hat. So we do play a role in epidemiology. We identify nosocomial infections and environmental pathogens. We do outbreak investigation. So in other words, if there are cases of salmonella, tuberculosis, Ebola, and things like that, as a microbiologist, we do play an epidemiological role. We also play a preventative role where we monitor sterilization and we kind of keep our finger on the pulse of infection control. We look at environmental cultures, and we also want to make sure that equipment such as the equipment that they use for bronch washes and bronchial alveolar lavages, we want to make sure that all of that surgical equipment is sterile, and so we monitor that. So we do play a role in preventing the spread of infectious diseases. And then another role that we play as a microbiologist is an education role, where we train hospital personnel in the handling of biohazards and other aspects of infection control. We educate other health professionals and ancillary personnel such as housekeeping and people like that, and teach them on outbreaks and isolation. So let's go ahead and talk about safety a little bit more in the microbiology lab. It is regulated pretty closely by the Centers for Disease Control and Prevention and also OSHA, which is the Occupational Safety and Health Administration. They have some really good websites, so you might want to refer to the CDC website, which is www.cdc.gov, for information that you might be looking up as well. We want to be aware of hazards in the laboratory, biohazards such as blood and body fluids, chemical hazards such as sulfuric acid or hydrochloric acid, and also we want to be aware of physical hazards, and that would be trip, slip, and fall hazards such as cords on the ground and things like that. Biohazards are, of course, of special concern in the microbiology laboratory, and remember, a biohazard is something that originates from a living thing. Biohazards in the microbiology laboratory include things such as biological specimens. And biological specimens are any specimen that originates from a living organism. We also have to worry about cultures in the laboratory. Bacterial, viral, and fungal cultures are all things that we need to be aware of and be cautious of in the microbiology laboratory. And there are several ways that you can come across a biohazard in the microlab, including breathing it in, that's called inhalation. You could actually ingest it, eating it or swallowing it. You could have a break in the skin, such as dry skin or a puncture wound, or you could have a direct inoculation where there's a puncture with a needle, or if you've punctured yourself with an isolation loop with a bacterial colony on it. Those are ways that people can be exposed to biohazards in the laboratory: inhalation, ingestion, a break in the skin, and direct inoculation. There's all sorts of disease risks in the laboratory, including bloodborne risks and also kind of the miscellaneous risks. Some bloodborne risks that we have to worry about in the laboratory would be things like HIV. That's the virus that causes AIDS. We have to worry about hepatitis B virus, hepatitis C virus, and then some organisms such as Neisseria meningitidis. Keep in mind that hepatitis B is probably a little bit more risky than working with HIV. HBV, which is hepatitis B virus, is actually 100 times more infectious than HIV. There are about 12,000 healthcare workers that are exposed to hepatitis B virus annually. Other disease risks in the laboratory would be bacterial infections from exposure to culture plates. We want to be very mindful of working with growing cultures. Also, you have to be very mindful of working with any type of viral cultures as well. Now, some things need manipulated in the biosafety cabinet or the hood, and that would include things like the respiratory viruses and funguses such as coccidioides. We always want to work with anything that is a spore former inside the biological safety cabinet. There are several methods that we want to follow in order to promote safety in the laboratory. One safety control method is following universal precautions. This is basically assuming that all blood and body fluids are infectious. These guidelines are issued by the CDC on how to handle hazards like contact with blood and body fluids. You have to have a good knowledge of the infectious state of all body fluids. Also, we want to follow standard precautions. Standard precautions means that we want to watch for slip, trip, and fall hazards, electrical hazards, and things like that. Things that we can prevent, some safety control methods that we can use to prevent exposure to infectious substances would be things like hand washing, and these are preventive tactics according to the CDC. Hand washing, wearing gloves, wearing face protection, and then how we dispose of sharps. Engineering controls are controls that isolate or remove a hazard from the workplace. So there are several types of engineering controls in order to make the laboratory a little bit more safe. For example, specimen containment. So in other words, we want to make sure that our specimens are in closed tubes, and that if we use an instrument, we can actually use a closed tube sampling instrument. Okay? We want to prevent aerosols. So whenever we're spitting something in the centrifuge, we always want to use bucket lids and make sure that we have the lid down and we never hit the brake and open the lid. We want to use vented hoods and biohazard cabinets. We want to make sure that the vented hoods provide negative air pressure, especially when you're working with acid-fast organisms. We want to safely use centrifuges. Like I mentioned, we don't want to ever use the brake on a centrifuge unless it's an emergency. We want to wear safety shields and cover our eyes, nose, and mouth. We want to do safe pipetting. We never pipette by mouth. That was kind of an old thing from long ago. We always want to use a mechanical way of pipetting. And then we want to use disposable equipment if we can, such as disposable slides, disposable tubes, and disposable pipettes. They're less expensive and they put us at risk a little bit less because we're only using that equipment one time. Work practice controls are often referred to as common sense practices. There are several things that we can do, or things that we can do to make our jobs a little bit safer. For example, we can frequently wash our hands, and we want to frequently wash our hands and wash our hands for a good 20 seconds. That's going to reduce the risk of putting anything in our mouths or getting anything that's infectious on our hands. We want to keep objects away from the face. We never want to smell cultures unless somebody has told you that it's safe to do so. There are certain things we don't want in the laboratory, such as food. We don't want any drinks in the laboratory refrigerator. We don't want any gum. There should be no eating or drinking in the laboratory. No smoking, no vaping, and things like that. We just don't want to expose ourselves to anything that could get in our mouths. We want to store personal items. We have the cubbies that you can store your book bags in, and we want to do that so we're not bringing those personal items into the contaminated laboratory. Any kind of spills need to be cleaned immediately with 10% bleach. Sometimes if you're working in a parasitology lab, we might use up to a 40% bleach to clean our counters and clean our spills. We want to decontaminate work surfaces, and again, that's going to be with 10% bleach both before and after lab or the alcohol swipes. And then, of course, we want to dispose of contaminated items. So things that are sharps need to go in sharps containers. Non-sharps need to go in the biohazardous trash. Things like gloves and lab coats, they're fine to go in the regular trash as long as they're not visibly soiled with body fluids or culture material. There's personal protective equipment that we can don in order to make our work environment a little bit safer for us to be around, such as gloves. You can wear latex gloves, but usually laboratories are going to nitrile gloves. You have to wear lab coats or gowns. Never leave your lab coat on. Always take it off before you leave the laboratory. And then finally, we have face shields. Anytime that you're working with direct specimens, you should be protecting your face and mucous membranes. Also, other things that you may want to do when you're in the laboratory is tie back your long hair. You don't want any of your ankles or skin showing, and we definitely don't want open-toed or open-back shoes. These are ways that we can prevent us being exposed to an infectious organism. The process used to recognize hazardous characteristics of organisms, now this differs from biological threat agents, but the process used to recognize hazards and characteristics of organisms is called biological risk assessment. All microbiology labs have to have an understanding of the risk groups that organisms fall into. The World Health Organization has divided organisms into 4 risk groups, and those are based on degree of infectiveness. So we've got things like Group 1, Group 2, Group 3, and Group 4. The higher the risk group, the more precautions you have to take in order to prevent exposure to these types of organisms. So most environmental organisms are found in Group 1. These are organisms unlikely to cause human or animal diseases. Okay. In Group 2, we've got our moderate to individual risk organisms. These organisms provide a low community or outbreak risk. Some organisms that fall into Group 2 would be Clostridioides difficile or Staphylococcus aureus. Group 3 organisms are organisms that are considered high individual risk but low community risk. They can cause serious harm to individuals but are not necessarily communicable. Some examples of organisms that fall into the Group 3 category would be Salmonella, HIV, and E. coli 0157H7. And then finally we have our Group 4 risk group. This is where the organisms have a high individual risk and high community risk. These organisms are communicable, and the treatment and prevention of these diseases is a little bit limited. Some examples of organisms that fall into the Group 4 category would be the smallpox organism or the smallpox virus and Ebola. Now, working in the microbiology laboratory, you may actually smell certain odors of organisms, and those odors can be helpful in the identification of certain organisms. However, you do not want to sniff any plates unless directed to do so by your instructor. The reason why is there could be fungus on those plates, such as coccidioides, or other things such as Francisella tularensis that could cause you to get very, very sick. And so you never want to sniff plates unless you are directed to do so by the instructor. However, you may notice odors emanating from your plates, and those odors can help you identify those organisms. Now, anytime you're working with plates that are growing fungi, you have to seal those plates immediately and also work with them in a biological safety hood with negative air pressure. You don't want any spores getting into the laboratory and ending up on your other culture plates. Now, there are some organisms that can be aerosolized, so you have to work with those inside the hood, and that would be Mycobacterium tuberculosis or brucellosis, or Brucella is the name of that organism. Okay, we want to wash hands frequently, avoid touching our nose, mouth, and eyes, and then cover all of our hangnails or dry skin or any cuts that we have on our hands with a bandage. Now, keep in mind, whenever you're working with a biological safety cabinet, a chemical hood cannot be used as a biological hood and vice versa. A type of containment barrier that protects the lab from aerosolized transmission of organisms is called a biological safety cabinet. There are several classes of hoods that contain aerosols. The classes of hoods are referred to as Class 1, Class 2, and Class 3. We happen to be working with a Class 2 hood in our student laboratory. Most microlabs have a Class 2 hood, and the hood is used for most infectious organisms, but the more serious infectious organisms require a Class 3 safety cabinet. The Class 3 safety cabinet is self-contained, ventilated, and has attached gloves for manipulation of culture. Remember, a chemical hood cannot be used as a biological hood and vice versa. Let's go ahead and talk about the biosafety levels. These are levels of protection for performing certain procedures. So for example, level 1 is when you're working with organisms not known to cause diseases in healthy humans. Here you have to wear your proper personal protective equipment. You need to wash your hands when working with these organisms and decontaminate surfaces. Some examples of organisms in the level 1 would be bacillus or other environmental organisms. In level 2 biosafety level, we are working with organisms that are a moderate hazard, and we're going to use those level 1 precautions, and then we're also going to limit employee access, and employees should get vaccines for certain organisms that are in the level 2 group. Some examples of organisms in the level 2 group would be Salmonella, HIV, and even hepatitis B. Then we've got the level 3 biosafety level. We are using the level 3 biosafety level when working with indigenous or exotic microbes. We always want to use negative air pressure, and these organisms have to be worked with in a segregated room. All windows and door seams have to be sealed. And some examples of the level 3 organisms would be Mycobacterium tuberculosis or St. Louis encephalovirus. Level 4 is when we're working with known dangerous and exotic microbes. Life-threatening organisms are the ones that are worked with in level 4. People that are working with level 4 organisms have highly restricted access to those rooms, and there should be potential signs posted that say that there are exotic and dangerous organisms being worked with in your laboratory. Sometimes you'll see people working with level 4 organisms kind of in those spacesuits, and also their rooms are equipped with particulate filters. Now, some examples of the level 4 organisms would be things like the Ebola virus, and then the hemorrhagic fever viruses such as Marburg virus, Congo Crimean hemorrhagic fever is another example of the level 4 organisms. Any substance that poses a significant hazard or a potential threat to human health or the environment is considered hazardous waste. Now, these substances require special handling, processing, and disposal. Anytime you're disposing of hazardous waste, you have to put it in one of the orange or red biohazard waste receptacles, and you will notice that it has the warning hazardous waste symbol on the receptacle. The Laboratory Response Network was designed to respond quickly to biowarfare threats. Infectious agents are divided into categories based on the severity if you're exposed to them. So the Laboratory Response Network was first developed by the CDC in 1999, and the network, like I said, responds quickly to threats of bioterrorism. There are 3 categories, including Category A, Category B, and Category C. The highly infectious organisms are in Category A, and that would include things like smallpox, anthrax, and tularemia. The moderately infectious organisms are in Category B, and that would be things like Q fever, brucella, and salmonella. And then finally we have Category C, which are the unknown or emerging pathogens, and some examples of organisms in Category C would be dengue fever and the hemorrhagic fevers. And speaking of safety in the laboratory, I want you to be familiar with some of these GHS pictograms and hazards. GHS stands for Global Harmonized System, which is a series of recognizable symbols for global recognition of all hazards, and they are universally understandable. So if you can see here, if you take a look at that bottom left example of that pictogram, you have a dead tree and a dead fish, and that is a symbol that universally is understood as being an environmental hazard. All right, so that was a little bit about safety and a little bit about the CDC, the World Health Organization, OSHA, ways that we can prevent exposures to infectious diseases. So let's go ahead and turn it around now and talk about the microbiological lab techniques. In the microbiology laboratory, we want to use what's known as aseptic technique whenever we're dealing with cultures. You have to understand that organisms are everywhere in the environment, and so as microbiologists, we want to avoid contaminants on our plates and on our slants and in our broths. Organisms of interest must be grown and studied in a controlled environment. Consider the optimal temperature and the optimal gaseous conditions for your organisms before you grow them out on culture. Aseptic technique is the cultivation of organisms under sterile conditions, and we want to prevent contamination. All culture and sensitivity tests must be set using sterile techniques. The microbiology laboratory must be controlled must be a controlled environment. So air currents have to be minimized, and that's so you don't have spores blowing onto your plate. So we want no fans in the room, we want all doors closed, and we want to keep our air currents to a minimum. We want to prepare slides and cultures using sterilized loops, sterilized probes, or applicator sticks that have been sterilized during our organism transfer. Plates should have the lid covering the culture media while not working directly with the culture. So you want to learn the proper way to hold plates, tubes, lids, and caps. Okay? Test tube caps should not be placed on contaminated surfaces, but instead should be placed in such a way that they're not becoming contaminated. Let's talk about the differences between disinfection and sterilization. So, disinfection refers to the use of physical processes to destroy vegetative microbes and viruses, but not those microbes producing endospores. So this is the process that removes a defined scope of organisms, but again, disinfection is not going to deal or not going to get rid of organisms such as bacillus or clostridium because they produce endospores. Some methods of disinfection would be chemical methods such as bleach solutions or alcohol. Another method of disinfection could be boiling or filtration. I don't know if any of you out there like to go camping or backpacking, but one way that you can disinfect water is by boiling it or filtering it. A disinfectant is a solution or process of cleaning inanimate surfaces. And again, we can use 10% hypochlorite, which is just bleach, or ETOH, which is alcohol, to disinfect surfaces. An antiseptic is a solution used to disinfect your skin, such as Purell or one of the alcohol hand sanitizers. Now, sterilization, this is a little bit different. This is the destruction of all living microbes, pathogenic or saprophytic. Sterilization includes the ability to destroy spore formers. So here, bacillus and clostridium will be able to be destroyed, and other living things will be destroyed as well. Now, sterilization can be accomplished by direct flame or intense heat, radiation, chemical or steam with or without pressure. The things that actually influence the degree of killing would be the number of organisms present, the time and temperature the organism is exposed to, the method of killing, the pH, and also if the organisms are living in a biofilm. All of those things affect how well something is disinfected or sterilized. One way that we can sterilize media to be used in the laboratory or sterilize things that are going to the biohazard bin would be by using an autoclave. An autoclave uses heat, pressure, and steam to sterilize whatever it is you're trying to sterilize. So the heat or the temperature of the autoclave is usually about 121.6 degrees Celsius. The pressure is 15 pounds per square inch. And the steam provides moisture. Your textbook says that something that is being sterilized should be sterilized for 30 minutes minimum. Okay? Autoclaving is the most effective means of killing microbes, including your spore formers, and those spore formers that I've mentioned before are bacillus and clostridium. Okay? The effective temperature is 121 degrees Celsius. The effective time is 30 minutes according to your textbook. Once the desired temperature is reached. So it actually has to come up to the proper temperature and then it sits there for 30 minutes. And then the effective pressure for an autoclave is 15 pounds per square inch. Let's talk about examination in the microbiology laboratory and the identification of microbes. Okay, this is known as the 5 I's. Inoculation. So in other words, we want to inoculate our culture specimen to the correct media. The second I is incubation. We want to use the correct temperature and atmosphere to incubate our cultures. The third I is isolation, where we thin out mixed cultures by getting isolated colonies. The fourth I is inspection or observation, where we make macroscopic and microscopic observations. And then finally, the fifth part, or the fifth I is identification, where we set up confirmatory tests that prove the organism's identity. That is known as the 5 I's in microbiology. All right, let's talk a little bit about culture media. And just so you know, your textbook has an excellent appendix in the back of the book that talks about all the different types of culture media, so you should be looking at that, okay? Some physical properties of media is they're either going to be liquid, semi-solid, or solid media. Liquid media would include things such as broths like nutrient broth and milks such as litmus milk, okay? Or brain heart infusion broth, and again, litmus milk. Some semi-solid medias would be things like motility media or gelatinase. And then some examples of solid media would be anything that's made of agar. That would include your blood agar plates, your McConkey agar plates, or even your chocolate agar plates. Those are the types of culture medias you're going to see in the laboratory: liquid, semi-solid, or solid media. Now, the functions of these media are several-fold, including basal media, which is basically basic. It is a good holding or banking media for organisms that we're trying to save. Then we have enrichment media, and some examples of enrichment media would be blood agar or Columbia nalidixic acid agar. We have selective media such as Columbia nalidixic acid agar or McConkey. We have differential media such as McConkey or XLD. And then we have biochemical media. That's the media we use to metabolically test our organisms. Let's go ahead and take a look at those different media types. So the first one I want to talk about is basal media. We don't really use a lot of basal media in a medical microbiology lab, but we do use it sometimes. But this media is very, very basic. It's not enriched at all. It usually includes such things as peptones, which is a polypeptide and an amino acid. Many amino acids bonded in a chain make it part of a protein. Okay, it contains peptones, meat-infused items, electrolytes, and agar. One example of a basal media, which is a basic media, is nutrient agar or nutrient broth. And you can see in that picture on the right, we've got a slant. The first tube on the left is a slant. That's a nutrient agar slant, which is basic. And then we've got two broths there, which are actually nutrient broths. And again, they're very basic. Agar, of course, is a complex solution of polysaccharides from the red algae known as Gelidium species. And we're going to talk about agar a little bit more in the upcoming slides. Enriched media contains complex organic substances like blood, serum, and growth factors. The top picture on the right there is an example of 5% tryptic soy agar with blood, and that has sheep's blood in it as what's enriching that media. It provides a source of nutrition for the bacteria that you're trying to grow. Enriched media allows for the cultivation of more fastidious organisms, such as the agar on the bottom right image. We've got chocolate agar, which does not contain chocolate in it, but it does contain blood cells that have been heated and lysed to remove or release the hemoglobin, and that's what makes it the color of chocolate. Blood, serum, and growth factors can be used by organisms for nutrition. Selective media is media that grows some things and then suppresses other things, okay? So it is useful for growing pathogens versus non-pathogens or normal flora. Some examples of selective media would be CNA or PEA, which are selective for gram-positives, or McConkey agar, which is selective for your gut organisms such as the enterics and some non-fermenters. Another example of a selective media is called mannitol salt agar, and we're going to be using that when we get to the Staphylococcus laboratory. Mannitol salt agar has 7.5% sodium chloride in it. Staphylococcus can survive that, but other organisms such as micrococcus cannot. So mannitol salt agar is selective for staph, while it does not grow other things. Okay, so in that respect, mannitol salt agar is selective. Differential media grows several types of organisms, but we can use it to determine the metabolic activities of these organisms. Okay? Components in the media that allow for differentiation would include things like dyes and pH indicators. A great example of a differential media is McConkey agar, where organisms that ferment lactose will turn pink. So an example of that would be E. coli, and then colorless organisms that do not ferment lactose are going to remain colorless, like I said. So some examples of colorless organisms that do not ferment lactose would be things like Proteus or Providencia. Okay, so we can take a look at our macaque agar and we can tell right away which are our lactose fermenters and which are our non-lactose fermenters. Biochemical media is used to determine fermentative or metabolic processes, such as gas production and also hydrogen sulfide production. These are highly specialized medias that determine enzymatic, fermentative, or biochemical properties of a specific organism. There are tons of varieties of these types of biochemical medias. The ones that are on the right in this image are triple sugar iron agar slants, and they basically show which sugars our organism will ferment and which ones they will not ferment, and then it also shows gas production and hydrogen sulfide production. Okay, there's a variety of these media types, and they're very expensive. They're usually about $3 to $7 a tube, and they should be used only when we're trying to identify an organism such as a stool organism like Salmonella or Shigella. So we don't use these on a daily basis because they are very expensive, but if we're trying to identify an organism that's not cooperating, sometimes the use of biochemical media is the way to go about doing that. So again, check out your media notes in your textbook in Appendix A at the back of your book. But we've got some examples of different media. So an example of a basal media would be nutrient agar or nutrient broth. An example of an enrichment media would be 5% sheep's blood. That's also known as BAP. We have enrichment and selective, and one example of that would be Columbia Callistin nalidixic acid agar, or CNA, that happens to be selective for gram-positive organisms. And CNA is also enriched because it contains blood, which supports the growth of those gram-positive organisms. We have got selective and differential media. Some examples of that would be macaque agar, also known as MAC, or eosin methylene blue, which is known as EMB. Macaque is selective and differential for gram-negative enterics, and it shows lactose fermentation versus non-lactose fermentation. And then we have biochemical medias, and there's a whole array of different biochemical medias or metabolic medias. One such example is triple sugar iron agar that contains lactose, sucrose, and glucose, and also H2S indicators. We also have indole broths, motility indole ornithine slants. We have cysteine trypticase agars or CTAs, which has virtually no meat or plant carbohydrates. You can actually add carbohydrates to that. It has a phenol red pH indicator, which denotes fermentation. Okay, many of these medias are pretty cheap, like our basal and enrichment medias are about 60 to 90 cents per plate, and then you get into those metabolic biochemical medias, and they can run the gamut from $3 to about $10 a tube. Now, live media is media that we usually use to culture certain viruses, okay? One example would be test tubes that contain rhesus monkey cells. Those are often called roll tubes. Other primate cells like human kidney cells or African green monkey cells can be used in live media as well. The tubes are inoculated with the virus, and then if you look at those roll tubes on a microscope, you can see that the cells start to swell. This is called cytopathogenic effect. If the cells start to swell, it means that they have been infected with the virus. Okay, so you can actually scrape out those cells and then use other serological methods to identify the virus that has infected the monkey cells. So let's go ahead and get you familiar with some of the media that is commonly used in student laboratory. Okay, we use a lot of different medias in student lab, but these are the ones that are probably the most common. We use a ton of blood agar plates. This is also known as trypticase soy agar with blood, okay? But basically a blood agar plate has 5% sheep's blood in it, and we use sheep's blood because it's cheap and it also suppresses other organisms that we don't necessarily want to see. We don't use human blood or rabbit blood except for in other cases, but this is 5% sheep's blood. They're about 90 cents per plate. Then we have CNA, which is Columbia colistin nalidixic acid agar. It is enriched and also selective. It is enriched with blood and it is selective for gram-positive organisms. It's the colistin and nalidixic acid that suppresses the growth of those gram-negative organisms. So CNA is going to grow things like Staphylococcus and Streptococcus, but then other things like your E. coli will not grow there. On the other hand, McConkey agar is selective and differential for gram-negative organisms. So your gram-negative organisms will grow on MAC because gram-positives have been inhibited by bile salts and neutral red. Okay, now organisms that are lactose fermenters are going to appear pink, and non-lactose fermenters are going to appear colorless. So McConkey is selective and differential for enterics or non-fermenters. There's a number of different isolation techniques that we can use in a microbiology laboratory. The whole idea of isolation is that we want to get colonies that are pure and not mixed with other culture organisms so we can do testing and get accurate results. Okay, so isolation is a method for microbial isolation. Okay, and there are several methods that we can use. The most common method of isolation is called the streak plate. Here we're using a streak plate to obtain isolated colonies. And what we'll do is we'll take a plate and we will actually streak it out 4 times, sterilizing in between each of the 4 streaks. And what that does is it thins out the population of organism and it makes it so we will get isolated colonies and no mixed colonies. Another type of isolation is called the pour plate. This is used to quantitate the number of organisms per mil. Sometimes it is used with calibrated loops. For example, we can use pour plates or the pour plate method for urine cultures when we're trying to determine the extent of an infection. A spread plate is used to quantitate the number of organisms per mil. A drop of bacterial suspension is placed in the center of the plate and spread out using a bent glass or metal rod. It's good for very, very dilute liquid cultures. We add a drop to the middle of our plate and then spin the plate with what's known as an L spreader. So let's take a look at streak plates and pour plates. So here's an example of a streak plate, and this is the technique we're going to use probably 99% of the time in student laboratory and in microbiology laboratories. In a streak plate, what we do is we put our organism in one quadrant of the plate, and then we take a Nicron loop and we spread it out, and then we actually sterilize our loop in between the streaks, and we come out 4 times, eventually thinning out that population of organism. You can see in the image on the top left of the image that we have isolated colonies. We didn't actually make those dots of colonies. Those colonies just appeared thinned out, and each organism is in a colony form because it has been thinned out by streaking from one quadrant to the next. We're going to use pour plates most of the time when we're trying to actually give the physician an actual number of organisms growing per mil. So we use calibrated loops, and the calibrated loops are usually a 1 to 1,000th of a mil or 1 to 100th of a mil loop. Okay, so what we can do is we could take that loop that has been calibrated and place it into our sample and then onto the media and streak it 3 ways all the way across the plate in 3 directions. So here we're not streaking for isolation. We're not doing a 4-streak method. Instead, we are streaking the sample all the way across the plate from the top to the bottom, 3 different directions. Okay, and then what we do is we incubate those plates, and then we count the number of colonies and multiply that times the dilution factor. So if I'm looking at my 1 to 1,000 plate and I've got 5 colonies, I could actually say there's 5,000 colonies growing per mil. So that's how we use pour plates, and most of the time that's used for urine cultures. Most human pathogens are mesophiles. These are organisms that prefer temperatures near body temperature. So mesophiles usually prefer 20 to 45 degrees Celsius, and human pathogens and human flora prefer 35 degrees Celsius. We've also got our psychrophiles, which prefer the cold. These are organisms that prefer negative 15 to positive 10 degrees Celsius. These are organisms that make their own internal antifreeze, so to speak. This would be things like arthrobacteria and some of the pseudomonids. Then we've got our thermophiles, which are the organisms that prefer hot temperatures. These are organisms that prefer 45 to 122 degrees Celsius. And these are organisms that you might find somewhere like the deep sea vents or the hot springs. One such example of a thermophile is Thermus aquaticus. When trying to culture our organism, we always have to be mindful of the gaseous requirements for each organism we're trying to culture. We've got organisms that are aerobic. These are organisms that grow in the presence of free oxygen. Some examples of aerobes would be Staphylococcus and Pseudomonas. We've got our anaerobic organisms, which are organisms that grow without the presence of free oxygen. And those would be things like Bacteroides or Clostridium. Then we've got our capnophiles, or our capnophilic organisms, and they prefer 3 to 10% carbon dioxide. These are usually our fastidious organisms such as Neisseria, Haemophilus, and our alpha hemolytic streps. And then we have our microaerophiles, which are organisms that prefer about 85% nitrogen, 3 to 10% carbon dioxide, and low oxygen, usually about 5% oxygen. And one such example of a microaerophilic organism would be Campylobacter jejuni. There are a number of organisms that are considered to be facultative anaerobes. For example, Staphylococcus and our enterics are facultative anaerobes. They actually prefer an anaerobic environment, but they do fine and grow and reproduce fine aerobically as well. So these are called our facultative anaerobes. So usually our facultative anaerobes are grown aerobically just because it's cheaper to do so in the laboratory. Then we've got our obligate aerobes, which are organisms that require oxygen for survival. One such example of an obligate aerobe would be Pseudomonas. And then we have our obligate anaerobes, which are organisms that require no oxygen. So we have organisms that are aerotolerant. They prefer no oxygen but can survive it. And then we have obligate anaerobes that actually hate oxygen. Then finally, for our last slide, you might be asking, well, how do we get those atmospheres? How do we create those atmospheres? Okay, some incubators are aerobic incubators, and other incubators actually have canisters of CO2 attached to them. But in our student laboratory, you can actually create those environments by using a jar, okay? And these jars can be made to contain carbon dioxide, or they can be made to extinguish oxygen, or they can be made to increase nitrogen. So we've got a CO2 atmosphere, and you can see that jar over there on the right. In a CO2 atmosphere, this is also known as a candle jar, we can use a commercial gas pack. We put that gas pack in the jar, and then we shut the lid, and what it does is it extinguishes all the oxygen and provides carbon dioxide. Back in the old days, you would put a candle in the jar and light it, and then when the candle went out, it produced CO2. An anaerobic jar is a jar that requires a commercial gas pack, and we want to remove all of the oxygen in the jar. We also want to make sure that our plates are pre-reduced, and what I mean by that is we put our plates in a jar and we take out the oxygen of the plates before we culture our anaerobes onto them. And then finally, we can create an atmosphere that is microaerophilic as well, where we use a commercial gas pack to create an environment that is about 85% nitrogen, about 5% oxygen, and about 3 to 10% CO2. So that is it for today's lecture. That is Unit 1. If you guys have any questions, please reach out to me.